is. So it's great to have with us here Miquel Vila and Ariadna Laguna, who I've joined myself this afternoon, where we're going to spend an hour just discussing a little bit about themselves and the exciting work they've done. And then we're going to have a sort of interactive session. I'm going to ask questions, and I hope you're going to also contribute questions, which we can discuss amongst ourselves. The idea is it's pretty informal. So there will be no slides unless we really need them to illustrate at some point. So I thought I would start by first of all just introducing myself. I suppose I ought to do that really. I'm a neurologist in the United Kingdom in Cambridge, where I work on clinical research as well as more basic research and translational research. But the main topic that we're discussing this afternoon is the work, as I say, that Ariadna and Miquel do around a substance called neuromelanin, which we're going to come to in a minute. But I thought it might be useful to begin with Miquel telling us a little bit about himself. So he's a very young man, as you can see. So I'm interested to know, Miquel, where you did your degree and then where you did your PhD. So where did you do your original sort of university studies and then your thesis?
Yeah, well, thank you. I'm very excited also to be part of this first research spotlight. I'm not that young, so I'm 51. If everyone wants to know, if you
that's
have
young
to know.
To me.
Yeah. Okay. So yes, I did my medical studies actually in Barcelona, the University of Barcelona Medical School, where I was interested in studying the brain on one hand, and on the other also I was interested in aging, in studying aging, the process of aging. So it seemed that these two topics could converge on the study of the diseases of the brain linked to aging, like Parkinson's disease. So after the medical studies in Barcelona, so I had the opportunity to go to Paris at the Salpêtrière Hospital to join the laboratory of Professor Miquel Vila. And then I did my PhD there under the supervision of Etienne Hirsch. And basically, during my PhD work, I studied the consequences of neurodegeneration on the functioning of the basal ganglia. Basically, it was at that time that it was quite exciting, because it was at that time that it was emerging the deep brain stimulation as a potential, as a treatment for Parkinson's disease. So my PhD work in Paris was focused on the how the lack of dopamine in PD patients may alter all the circuitry of the basal ganglia that finally leads to the motor symptoms. And basically,
so this was in the 1990s then, Miquel. So this is when deep brain stimulation really came in probably 91, 92.
Yeah,
so
it was the 1990s. So we had there like a multidisciplinary group with neurosurgeons, neurologists, basic scientists, neuroanatomists, and electrophysiologists to try to define which one could be the best target for the brain stimulation. And it turned out to be mostly the subthalamic nucleus.
So but the Salpêtrière is very, very famous for Parkinson's disease. I mean, apart from all the great researchers, of obviously which you were contributing your PhD, I think that's where Charcot was in the late 19th century. And it was Charcot that gave Parkinson's its name, Parkinson's disease, I believe.
Yeah. Actually, it's considered the Salpêtrière where the modern neurology was actually born in hands of Charcot. I was there a little bit later than that, but not that much. And then I was interested to study, to continue after my PhD, not the consequences of cell death, but rather to go to study the actual cause or mechanisms or cell death, to go to the root of the problem, to see if it could be possible to try to stop or delay or even prevent a cell death. And for that, I moved to New York. I joined the lab of Dr. Serge Borsky at Columbia University.
Who's very heavily involved with the WPC, I should add.
Yeah, actually, this is the movement disorders unit that was led by Stanley Fan at that time. So there, I really was focused on the molecular mechanism of neurodegeneration, and I spent there several years, first as a postdoctoral researcher, and then as in a faculty position as assistant professor.
So when did you come back? So where are you now? Miquel, where are you?
Yeah, so after that, I had the opportunity to go back to Spain, to go back to Barcelona, to my hometown, to develop a new research lab at the Vall d'Hebron Research Institute, which is part of the Autonomous University of Barcelona. So I'm still here. So
how
now
many years ago did you come back to Barcelona?
It was 15 years ago. Actually,
15
it was 15 years ago right now.
So your interest in Parkinson's was sort of generated during your clinical work with an interest in the brain and aging, and then your PhD took you right to the heart of it with Yves Agid and Etienne Hirsch in Paris. And since then, you've never deviated from that course of trying to understand what goes wrong in Parkinson's disease.
Yeah, that's it. It's quite. I think it's quite exciting. Quite interesting, as we will discuss. But I don't see patients. I actually don't see patients. I'm focused on basic research.
Yeah, great. And Ariadna, I mean, tell us a little bit about yourself. How you know, where you did your earlier studies and how you ended up coming to work with Miquel.
Sure. So I am also born in Barcelona, and I also did my undergraduate here at the University Pompeu Fabra. And in my case, I studied human biology. So I'm a basic researcher. I have a basic researcher background. So I did my PhD here in Barcelona at the Center for Genomic Regulation, where I was basically working on the genetics of Down syndrome. So I was studying how gene dosage, gene balance was affecting the development of the brain and giving rise to the different neurological alterations in Down syndrome individuals. So that's how I became interested in neuroscience. And actually, I must say I became fascinated by neuroscience. So when I finished my PhD, I decided I would continue in the field. And then I looked for a European research center that was international, like very well internationally known, and that could give me like a better training. So then I decided to move to Sweden, to the Karolinska Institute. And that's where I did my postdoctoral studies. And I joined the lab of Professor Thomas Pellman, who was at the time very well known for his studies on dopaminergic neuron development and specification.
He's also the secretary of the Nobel Committee, isn't he?
Yes, exactly.
So useful person to know.
Yeah, I'm not expecting to get the Nobel Prize, but at least it was fun to be close to the Nobel Committee for those years and to be able to attend all the seminars when the Nobel laureates came to Sweden. Yeah, that was very fun. So in his lab, that's where I got to know Parkinson's disease. And by first studying the development of dopaminergic neurons, then we became interested in what function or what would the role be of transcription factors that were important for the development of these neurons that were later also expressed in mature adult neurons. So we wondered whether this could be actually involved in the pathology of Parkinson's disease. So we basically did a lot of mouse genetic studies looking at the function of these transcription factors in adult neurons, and realized that they were actually important, and ablation of these or dysfunction of these transcription factors was actually affecting the function of the neurons and giving rise to...
So the main work was, so it's quite interesting contrasting your two approaches. So Miquel was interested very much early on in why cells die as you get older. You've gone to the other extreme and said, you know, what makes the system develop in the first place? And are there problems in what normally happens in development, which sort of recapitulated or fails and leads to disease, if I've understood it?
Yeah, definitely. That was the situation. Yeah. So when I finished my postdoc, I decided I wanted to go back to Spain and contribute to the science that is done here. So I moved back to Barcelona and looking for a lab that was doing research on Parkinson's disease. I ended up joining Miquel's team and participating in this Neuromelanin exciting project.
So when did you come back to your home?
That was end of 2014.
2014.
Yeah.
Okay, so very interesting, slightly different paths you took to end up studying a sort of problem, which I suppose is very useful for people to hear, that actually approaching Parkinson's disease and trying to understand aspects of it, people from different backgrounds can bring different areas of expertise to try and solve a common problem. So both of you, if I've understood it correctly, seem a little bit obsessed with this thing called neuromelanin. So most of us will know a little bit about melanin because we would often come to Barcelona to try and stimulate our melanocytes so we could produce a bit more melanin and look a bit, a suntanned. So what is, I mean, Miquel or Ariadna, what is, perhaps Miquel could start us off. I mean, what is neuromelanin, and is it very different to the melanin we find in our skin? And why are you interested in it?
Yeah, but actually neuromelanin is quite similar to the melanin of the skin. That's why it's called neuromelanin. It's like the melanin of the skin, but inside a neuron. So that's why it's called neuromelanin. And basically, we can interested because neurons that have neuromelanin are the ones that preferentially degenerate in Parkinson's disease. So you find neuromelanin in a specific type of neurons that are the ones that produce a specific neurotransmitters like dopamine and noradrenaline. You only find neuromelanin inside these neurons. So neuromelanin is a pigment. It's actually a pigment similar to the melanin of the skin. Is a brown dark pigment. That is
this what gives the, just sorry to interrupt, I mean, is this what gives the name the substantia nigra? Because when people say that, it's the sort of black substance. Yeah. I mean, is that the neuromelanin we're looking at there, and which people are talking about when they're talking about the substantia nigra, which some people would hear is where the dopamine cells which die in Parkinson's reside? Is that right?
Yeah, actually the cells that accumulate more neuromelanin in the brain are the ones from the substantia nigra, and that's why, as you say, it's called substantia nigra. That means, as I say in the blog, black stuff. So it really means that because when you cut a brain, you can see with the naked eye, you can see this region, the substantia nigra, without any microscope. And the interest is because then the neurons that have the highest levels of neuromelanin in the brain, that are the ones from the substantia nigra, are actually the ones that preferentially degenerate. So that's why we became interested in this particular pigment.
And whilst it's just in these particular nerve cells, which are obviously susceptible to dying off in Parkinson's, I mean, how widely found is neuromelanin in other animals, in other species? I mean, is this something unique to humans, or do all animals have this? If we look for neuromelanin in other neurons and other nerve cells in rats or monkeys or whatever?
Yeah, well, actually, all humans produce neuromelanin. All of us produce neuromelanin. So neuromelanin, it's first produced and becomes visible by the age of 3. And then because neurons did not develop a system to degrade or eliminate this pigment, so then neuromelanin builds up inside the neurons with age. So that's why we accumulate more and more neuromelanin with age. And in the case of the substantia nigra, it becomes so high the levels of neuromelanin that where you can see then with the naked eye. And what is particular, that although we know from more than 100 years that neurons with the highest levels of neuromelanin are the ones that die in Parkinson's disease, in the substantia nigra. If we go back to the Salpêtrière Hospital, it was the PhD thesis of a student there, Konstantin Tretiakov, who cut the brains of several Parkinson PD patients and also of healthy controls, and who saw by the naked eye that the substantia nigra disappear because of the loss of the neurons that have neuromelanin. But although it has been known for 100 years that the neurons that have neuromelanin are the ones that disappear preferentially in Parkinson's disease or degenerate preferentially, the role of neuromelanin is not well known because, as you mentioned, neuromelanin is found mostly exclusively in humans, especially at those levels that are found in the human brain. So basically, in other animals, like the animals that are commonly used as experimental models in research, like rodents, rats, and mice, they do not have neuromelanin at all. So this factor that is so important for Parkinson's disease has not been properly studied because for the simple fact that animals that are used in research do not have neuromelanin.
And even if you look in very old animals, I mean, I don't know if you know the answer to this, Ariadna. If you look in very old monkeys, for example, which we often, I mean, it's obviously not experiments we do, but there will be aged monkeys in zoos and such like that die. Do you find neuromelanin in old monkeys? Is it just because you know we live to such an old age, and most of the work done in the lab would be in young animals? Is it just if you could keep animals alive for long enough, would they all develop it, or is it really just unique to humans?
Ariadna, do you want to answer, Ariadna? Or?
Yes. Well, you know more about primates, no, but in all primates, depending on the species, one can find neuromelanin as well. But in the case of rodents, you know, rats and mice usually live around 2 years. And we have been looking at that, and we haven't found neuromelanin. We haven't seen it by eye or under the microscope. You might see something similar to neuromelanin when you look under the electron microscope, but very, 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 very tiny granules of this pigment, never reaching the levels that one can see in the human brain.
Okay. So I'm just going to remind people, if you do have questions, don't forget to put them in the chat, because obviously we can then ask our experts here to try and answer those. So if I just stick with you, Ariadna, I mean, is this a good thing or a bad thing, neuromelanin? Having read several of your papers, I can't decide whether this is a good thing, because neuromelanin mops up and stops the cell from dying by protecting it, or is it a bad thing because the neuromelanin actually is promoting problems within the cell and killing them off? So I'm not sure whether neuromelanin is a good thing or a bad thing. I don't know if you could perhaps help us answer that.
That's a good question. That's a good question, and we are trying to find the answer for that. So what we know is that at least in the beginning, it might be a good thing, because it might be chelating toxic species or oxygen reactive species and other things that the cell cannot degrade, and it might
so it sort of soaks them up. It sort of takes them in like a sponge.
Yeah, it might be like a waste, like a collection of waste, and then and that might be beneficial for the cell. But at some point, it seems that when the cell gets too crowded and there is too much neuromelanin, then it starts interfering with the normal cellular function. So it starts, and then you start having problems with like mitochondrial function, proteostasis, and other like very basic homeostatic processes. And then it's when it might become bad. Like it might become a bad thing.
And so things like, just whilst I'm on it, and then I'll come back to Miquel a little more about how neuromelanin could be involved with Parkinson's disease. I mean, there's always been this question in Parkinson's disease, you know, the alpha-synuclein, which is obviously the protein which most people will think of with it, and it accumulates, and then you get these Lewy or Levy bodies. And there's always been this discussion, is this a good thing or is this a bad thing? So is it an attempt by the cell to put all the protein together, which is bad, and put it in a single dustbin and hope it stays there and doesn't cause problems? Or actually the Lewy body itself becomes a problem? So is neuromelanin a bit like that? And how does neuromelanin relate to this Lewy or Levy body? Are they separate? Do they talk to each other? Are they, I mean,
Yeah, actually, neuromelanin, as we discussed, accumulates with age. So what we have found, basically, the problem that we discussed before is that if we want to understand what's the potential role of neuromelanin, since we do not have any experimental animal model that produce neuromelanin at such a high levels as humans, so we had to create our own model. So we had to create our own rodent model that produced neuromelanin. So by genetic manipulations, we were able actually to produce rats that produce neuromelanin.
So just explain that, Miquel. So how exactly did you do that? Because if the rats don't do it and we get it with aging, did you artificially age the rats in some way?
Yeah.
Or how did you manage to get them to produce neuromelanin in their brains?
Yeah, actually by accelerating or by boosting the metabolism of dopamine, because we know that neuromelanin is produced through the oxidation of dopamine. Yeah. So then what we did is that we actually accelerated dopamine oxidation, basically by overexpressing one enzyme that is the one that is known to produce the melanin of the skin. So basically we put the enzyme of the production of melanin of the skin inside of the brain.
So this was a little, so you had a little virus in which you attached this enzyme, and then you injected it into the brain of the rat, into a specific site, presumably into the dopamine cells, so they then could start producing.
Exactly. Using this like gene therapy, using the virus to introduce a gene inside these cells. So we were able to produce neuromelanin. These animals started to produce neuromelanin that accumulated with age up to levels as an elder human brain. And what we found is that, as Ariadna mentioned, that as neuromelanin accumulates above a certain threshold of intracellular neuromelanin accumulation, then these neurons started to have a dysfunction, to be dysfunctional, and started to have an impaired dopamine release. And one of the things that we could see is that they will start forming this Lewy body-like structure. They were forming Lewy bodies that you were asking. So basically, these cells started to become dysfunctional, and finally they degenerated. And these animals actually develop all of the phenotypic features typical of Parkinson's disease, like motor dysfunction, downregulation of the phenotype of the dopaminergic phenotypic markers, impaired dopamine release, and then they appear the formation of Lewy bodies.
So basically, you got to this whole hypothesis by virtue of the fact that with aging, neuromelanin accumulates. Neuromelanin accumulates in the cells which are particularly vulnerable in Parkinson's disease. And to sort of, I mean, so some people could say, well, that's just a description. You know, we just see that Parkinson's disease happens when you're older. Neuromelanin happens when you're older. They're not related. You can sort of say that they occur in those cells. It might just be a byproduct of those cells. But you, by these experiments, have now started to demonstrate that neuromelanin isn't just something you observe in these cells, and with aging, it seems to be an integral part of what might be going wrong in Parkinson's disease through your system. And when you inject this virus, how do you make sure it stays in the dopamine cells, or does it go into all the cells in the area, but it's only the dopamine cells, by virtue of the fact they produce dopamine, that develops this pathology with the neuromelanin?
Yeah, actually it goes to all kinds of cells, but because neuromelanin is formed by oxidation of dopamine, only the cells that have dopamine actually that will generate neuromelanin.
Yeah. Actually, it was amazing. Like when injecting the virus, we only injected it in one side of the brain, and when we cut the brain of the rat, you could very nicely see one side of the brain, the substantia nigra, that was like drawn, while on the other side it was completely white. Okay. There was nothing.
So there's a question which has come up here, which I'm going to ask, which links also to another question which I have, which actually is quite interesting because it goes back to Miquel's PhD studies in some ways. So there was a story 25, 30 years ago that L-DOPA is itself toxic to cells. So there was this story going that one of the reasons people develop complications further down the line with Parkinson's disease, having had L-DOPA for some years, is actually L-DOPA is killing off the cells. And then there were studies done which in looking at cells in a dish in the lab, that if you gave high doses of L-DOPA, you could kill cells. And then at the, I think it was Yves then said, actually, L-DOPA is a bit neurotrophic. It actually encourages the growth of cells. It's not toxic. So a question I have for you is, if oxidation of dopamine, so when you take dopamine and it changes its form, this oxidation, it takes oxygen on and becomes a bit toxic and works with neuromelanin, is taking L-DOPA a bad thing because that's promoting that process? That's the first question I have. And then the second question, which relates to a question we've had posted, is how does neuromelanin, dopamine, and synuclein talk to each other? Because most people would say the problem in Parkinson's disease isn't really neuromelanin or dopamine, it's the accumulation of alpha-synuclein. So how does alpha-synuclein fit in with your story? So what role does L-DOPA have in this? Is that a bad thing to be taking? Because lots of people on this call and listening to this subsequently be saying, well, I take that drug. Should I stop taking it because it's a bad thing? Which I would not encourage, but it'd be useful to hear your comments on that. And secondly, how does your theory fit in, or this hypothesis about the crucial role of neuromelanin fit to what we know about alpha-synuclein?
Yeah, so basically regarding L-DOPA, L-DOPA is a precursor of dopamine, so it's a precursor of neuromelanin also. But Actually, when I work at the laboratory of Vivagit, there was someone else in the lab, Gustavo Murer, who tested this hypothesis whether levodopa was toxic or not. So he treated rats for 6 months with levodopa, and actually it was not toxic at all, even if you had an increased dopamine oxidation. But these animals, importantly, they do not develop neuromelanin. Even if you increase the levels of levodopa, you increase the levels of dopamine and oxidized dopamine, they did not produce neuromelanin. That for us is an important point, because when we wanted to develop this animal model that produced neuromelanin, we didn't know first how to do it. I mean, what do we know about the synthesis of neuromelanin? So it is believed that it's a spontaneous oxidation of dopamine, that you do not need any enzymatic intervention. So we thought that maybe just by increasing levodopa, by increasing dopamine levels and dopamine oxidation, you may get neuromelanin. But actually, if you do that, you don't get neuromelanin. If you just increase dopamine levels and dopamine oxidation in animals. So only when we overexpress this tyrosinase enzyme, then we got neuromelanin.
So that is an enzyme which converts dopamine into melanin, neuromelanin, is it?
Yeah, actually it oxidizes dopamine into dopamine kinones that then convert into neuromelanin.
Okay.
So and basically there is a nice historic paper by Stanley Fan who describes a little bit the history of the discovery of levodopa by George Kochias. And apparently what he mentions is that Kochias initially he used levodopa because he thought that maybe what Parkinson's disease patients needed, it was to increase the levels of neuromelanin. Because in PD patients, you had a loss of the substantia nigra, meant you needed to restore this neuromelanin. So apparently, according to Stanley Fan, so Kochias started giving levodopa to try to see if it could increase the levels of neuromelanin in these patients. So it did not increase the levels of neuromelanin, but rather had the effective effect of increase of dopamine. And then regarding
we actually just have confirmation. So Charles Messel in the chat has just said he's also found in a recent range study that treatment with L-dopa prior to the administration of chronic MPTP, which is a toxin obviously for dopamine cells, was neither protective nor toxic. So I guess it sort of fits in with what you're saying. So sorry, carry on, Miquel.
Yeah, so maybe for the synuclein, do you want to discuss that, Ariadna?
Yeah, I can.
The link between synuclein and neuromelanin?
Yeah, sure. This is also another question that the Charles had on the chat. So how could one differentiate between the toxic effect of neuromelanin and the synuclein one? So actually, this is also a question we had. And this is something we addressed in our paper. And the way we did that was that we actually took advantage of a knockout alpha-synuclein animal model. So it was a mouse that did not have alpha-synuclein. So in this animal, we, using again the same virus strategy, we overexpressed this tyrosinase enzyme in the brain of the mouse. And again, we saw the production and the accumulation of neuromelanin. And as Miquel said before, we also saw the formation of these Lewy body-like type structures that were positive for P62 or ubiquitin, for example, which are known markers of Lewy bodies. However, in these animals, there was no alpha-synuclein. And when we stained for those Lewy body-like structures, obviously there was no alpha-synuclein staining. So at least in our models, the absence of alpha-synuclein had the very same effects in terms of the toxicity of neuromelanin.
And did those animals also have behavioral deficits? So it wasn't just, so they developed neuromelanin and inclusions, and they also then the cells were dysfunctional, and then they died, and the animals had a phenotype, had behavioral problems.
Yeah, they had exactly the same phenotype in mice. Yeah. But
so do you think there are two pathologies then in Parkinson's? So is there an alpha-synuclein mediated sort of what we'd call a proteinopathy, and then there's an independent process which is around neuromelanin mediated inclusion body formation? I mean, it comes back to the question I was asking. Do you think these two systems talk to each other?
Yeah, well, it may be the same process. Basically, what we were surprised to find out is that okay, with accumulation of neuromelanin, we could see the formation of these alpha-synuclein positive inclusions in our rats. And there was some studies by Glenda Halliday, she described in human brains that apparently alpha-synuclein can translocate to the neuromelanin pigment, and especially the lipidic part of the neuromelanin pigment, and that the neuromelanin could trap somehow alpha-synuclein and start the formation of the Lewy body. So what we were surprised.
So what you're saying is alpha-synuclein could accumulate in any cell. I mean, it doesn't. It accumulates in some cells and bugs, and that would be independent of neuromelanin. But what neuromelanin does is it speeds up that process. It promotes it because it has an affinity for alpha-synuclein, which allows the alpha-synuclein to come in, alpha-synuclein can bind to it, and so the inclusion bodies and the toxicity can be magnified by virtue of having neuromelanin.
Yeah, it's possible, but actually it's not only synuclein. The Lewy body-like structures that we found in our animals, they had synuclein, but they had other components of the Lewy bodies like P62 or ubiquitin. What we were surprised to find out is that when we asked the question, okay, the toxic effect that we see in our animals that produce neuromelanin could be mediated by alpha-synuclein, and that's when we performed the experiment that Ariadna mentioned, that we remove alpha-synuclein from these animals, and we were expecting to see maybe a reduction of Lewy body formation and cell death. But we actually saw the same degree of Lewy body formation. But those were Lewy bodies without alpha-synuclein, because alpha-synuclein was not there, but they were still P62 positive, ubiquitin positive. I don't know if you can call that still a Lewy body if it doesn't have alpha-synuclein. But it seemed, according to our experiments, that maybe, although when alpha-synuclein is present, then it contributes to the formation of the Lewy body, but maybe when alpha-synuclein is not there, actually it's not required for the formation of the Lewy body.
And did you see any difference in the kinetics of what you saw? So did it happen more quickly if you had a mouse or a rat with alpha-synuclein in which you put your your enzyme? So did the pathology develop more quickly and the problems develop more quickly in those rodents? Or
no.
If you had knockout, you didn't have any alpha-synuclein. So they both developed the pathology at the same time with the same deficits, irrespective of whether alpha-synuclein was there or not.
Yeah, exactly the same. Now we are doing the opposite experiment. So what we are doing is we are overexpressing alpha-synuclein in animals with neuromelanin to see if this can accelerate the process. So in this case, it seems so, if we overexpress alpha-synuclein.
So that's very interesting. So I'll come back a minute to that, because obviously one of the questions which naturally arises from this is how we can take the information which you have generated and your model to talk about therapeutics, but we'll come on to that in a minute, because one of the areas which you discuss in some of your papers is, and is of course very topical at the moment in POG disease, is this idea of inflammation in the brain. So, the idea that whatever goes wrong, and now we're having to rethink from what you've said, because we would sort of say alpha-synuclein leads to toxicity in the cells become unhappy, the cells die. And then there's inflammation around the sites of pathology or at the sites of pathology. And one of the questions in Parkinson's disease has always been, is that simply a mopping up process? The cells die, inflammation comes, like when you cut your skin, you get inflammation and it just turns up because you've injured yourself. Or is inflammation itself driving the disease process? So I wondered what role you felt your model or your mechanisms had for inflammation. Is neuromelanin, if it's released from cells, for example, generate much more of an immune response than if you just a cell that dies without it? How does inflammation factor into your theories?
Yeah, it's exactly as you mentioned, because once the neurons that contain neuromelanin, they start to degenerate, so then neuromelanin becomes extracellular, that it's released from the cells that are dying, and then you have an activation of microglial cells that they try to eat.
So these are the immune cells in the brain, the microglia.
Yeah, and they try to remove this neuromelanin from the brain, this one that has been released from the neurons. So you have a strong activation of inflammation following the release of the neuromelanin from the cells that are dying. And now we are studying whether this inflammation can contribute also to the progression of the degeneration, that you have so much inflammation that these inflammatory cells, they start by eating the neuromelanin that is released, but maybe they are so active that they can also eat the neurons that are still there. So that could
but you don't get the Lewy bodies in the microglia. So although you can eat the neuromelanin, they can't make dopamine and oxidized dopamine. So they don't themselves die off as a result of this process.
No, not at all. Because in contrast to neurons cannot degrade neuromelanin, but microglia can degrade neuromelanin. So they have the machinery to degrade neuromelanin. So they can dissolve the neuromelanin.
Okay. So if I've understood the theory correctly, and so the hypothesis would be that as we get older, we all produce more neuromelanin in our brains, particularly in the sites that are making things from L-DOPA, if you like, so dopamine and noradrenaline or norepinephrine, so certain cells in the brain. And with time, that then leads to a pathology because the neuromelanin and the oxidized dopamine, with or without alpha-synuclein, causes the cells to dysfunction, die. You then get inflammation, which then sort of amplifies that process, and you go on and develop POG disease. So there's two questions which I'd have to that. One is, if this is true, why is it we don't all get Parkinson's disease? Because we're all getting older. That's one thing I can say with absolute certainty. We're all developing neuromelanin in our cells. So why is it some people get POG disease and others don't? And then after that, I want to discuss, in the last third of this hour, a little bit about how we can tackle this therapeutically. So why is it not everybody gets Parkinson's disease if this is a normal process? So if I was to take, just to sort of put it into context, if I was to take 70-year-old brains and cut them, would every single one of those brains have neuromelanin in them to roughly the same level? Or is it that some people get to 70 with very little neuromelanin and some people get to 70 with lots of neuromelanin? So how much does it vary as we get older, and why do only some people get POG disease as a result?
Yeah, well, I can start, Ariadna, but then you can continue that. Indeed, what we found is that you start having this pathological effect of neuromelanin when you reach a certain level of neuromelanin accumulation, above a certain threshold of neuromelanin accumulation. So you start seeing this pathological effect. And actually, we saw that in our animal model, in our rats. But then we took brains from healthy aged individuals and age-matched Parkinsonian patients, and we also analyzed the levels of intracellular neuromelanin of these brains. So we found that the brains from healthy individuals, the levels of neuromelanin actually were below this pathological threshold. And the brains from the PD patients were above this pathological threshold. So somehow the patients that had Parkinson's disease reached earlier this pathological threshold of neuromelanin accumulation. And actually, we took also brains of subjects that they were supposed to be healthy controls, and that happens sometimes. So then you cut the brain, and they do not have Parkinson's disease, clinical Parkinson's disease, but then you cut the brain using it as a control, apparent control, but then you found that they have already Lewy bodies. So these cases are called incidental Lewy body disease. So, and are believed to be like pre-PD stages, very early stages of PD, that maybe if they had lived longer, they could develop PD. So these incidental Lewy body disease brains, they also had these intracellular neuromelanin levels above this pathological threshold. So that means that the people who have an accelerated formation of neuromelanin and reach neuromelanin levels that are above this threshold, so will be the ones that generate neuromelanin, and maybe Parkinson's disease, I mean, and maybe all of us will finally develop Parkinson's disease if we were to live enough to reach these pathological levels of neuromelanin. Okay. So I don't know, Ariadna, if you want to add something.
Yeah, maybe just to add that, so as you said, Roger, like the big question is why some people develop or like reach these levels of high levels of neuromelanin before the others. So an obvious possibility is like different genetic backgrounds, like, and different exposure to environmental toxins or different lifestyles. We know like everything influences the function of our body. So one thing that we are looking now is we are looking at brains from people with Parkinson's with different genetic mutations, and then trying to look at the levels of neuromelanin in those brains to see if those were if those are higher or lower, and also trying to look at brains from subjects that were carrying mutations in these genes associated to Parkinson's disease, but that didn't manifest the disease yet. So trying to see if those, let's say, prodromal or pre-diagnosis PD subjects would also have high levels of neuromelanin or not. And of course, another issue, another question is because we have seen that the enzyme tyrosinase seems to be so important for the formation of neuromelanin, we are also looking at people having mutations in this enzyme and trying to see if mutations in the enzyme that correlate with the function of this enzyme actually have something to do with the levels of neuromelanin, and if this could also be an explanation for these people reaching
presumably
levels
the people
of
who have abnormalities in that enzyme will be people who are sort of albino, people who don't have normal pigmentation. Yes. Would that be right?
Yeah, maybe. Miquel, you can take the albino part, which is your favorite.
So just before we do that, I just want to ask you another question, Ariadna, because you've got this beautiful model. So you've now got in the lab a reproducible model where you can produce neuromelanin and Parkinsonian-like pathology whenever you want. So one of the questions, just before we come on to the genetics, if you like, around albinoism and genetic variants and whether that plays into it. I mean, people are always very interested in environmental toxins, as you said, Ariadna, but things like, insecticide, pesticides, paraquat, these type of things. So if you take your animal model, you may not have done this, and obviously there may be issues around licensing and what people allow you to do, but expose them to known toxins like that. Does that promote the pathology quicker? So if I came along with a pile of paraquat and gave it to your animals, would they quickly develop toxicity in the brain, lose cells, which would sort of suggest actually environmental agents could play out in this system through this mechanism with neuromelanin?
Yeah, you're absolutely right. This is actually something we are currently doing. So we have exposed these animals to MPTP, like a very well-known neurotoxin that is widely used as a Parkinsonian model as well. And we haven't got the results yet, so I cannot actually tell you what happens, but we have done this. And of course, the hypothesis would be that there would be some sort of additive effect. And we are also looking like other types of environmental factors that could also modulate or affect the development of the pathology. And we are looking at the microbiota, for example, which is something that is a very hot topic now. So
also, so this is all the bacteria in the gut, and how the flora and fauna in your gut influences your brain, and obviously, as you say, lots of links between this microbiome and how it plays out in Parkinson's disease.
Yeah, absolutely. So now we have a good model where to test if by manipulating, for example, this gut microbiome, if the pathology is influenced either in a positive or a negative way. This is something that could be addressed using this model.
So beautiful way of studying these sort of environmental and sort of distant parts. So Miquel, coming back to you and genes. So I mean, obviously there's lots of genetic studies which link genes to POGSI. So off the top of my head, and it's always expanding that list, I can't think of any particularly that link to the enzyme involved with making neuromelanin. But you were saying, looking at people who have genetic defects in that enzyme, so people who get forms of albinoism, do they have an increased risk of getting Parkinson's disease, or?
Well, actually, it was mostly in relation to try to understand how neuromelanin is synthesized in the human brain, because it is not clear, but it's supposed not to be mediated by this enzyme tyrosinase, although these enzymes appears to be expressed at very low levels in the brain. So what Ariadna was referring to, I believe, is that to try to understand whether tyrosinase may participate to the formation of neuromelanin. So we can study this in subjects with albinism that may have a specific genetic alterations in tyrosinase. There is only one variant that is published, one variant of tyrosinase that increases the risk of Parkinson's disease. That has only been described by one group.
Okay, so now we've got this beautiful model, and we've sort of understood the sort of principles and what you've discovered. So I guess, you know, the big question will be, so what? I mean, that's great to understand the pathways and the role that this may have, but how can this be changed into something therapeutically? So obviously you could say, well, if it causes more inflammation, and as one of the question here says, if we block inflammation, that might be a good thing. But you could say that's a bit downstream of the problem we've got. Is there anything we can do about manipulating the formation of neuromelanin to help people with Parkinson's to slow down the disease course? And it comes back to a sort of earlier question and discussion is, if you got rid of neuromelanin altogether, so you had some clever way of washing it out of the system, would that be a disaster for the cell, or would that be the eureka moment we've all been waiting for to cure Parkinson's disease? Or should we be looking at drugs that promote more neuromelanin to try and protect more cells? So what sort of therapeutic approaches should we be thinking about, and how do you get it right so you get the right level of neuromelanin to make sure it's doing the right thing?
Yeah, actually, based on the results that we got from these animals, it would seem that to have a therapeutic effect, what we would need to do is to reduce the levels of neuromelanin, but not to reduce the synthesis, because as we discussed before, maybe the formation of neuromelanin itself is initially protective because it detoxifies some oxidized dopamine elements that may be toxic to the cells. So the best approach could be to be able to eliminate neuromelanin or to prevent the accumulation of neuromelanin, not the synthesis, but the accumulation, to be able to promote somehow the degradation or the release of neuromelanin. That's actually what we did in our article that we published a couple of years ago. One of the proof of concept approaches that we use is by gene therapy. We promote, because neuromelanin, we know that the cell tries to eliminate the neuromelanin. So what it does, it accumulates neuromelanin inside what is called autophagic vacuoles to try to eliminate neuromelanin. But because neuromelanin is very insoluble, it cannot be degraded. So what we did is that we overexpress a factor that is a promoter of autophagy, is a master regulator of autophagy, that among other things, it helps the cell to clear the cell, to release outside of the cell these autophagic structures to try to clear the cytoplasm. So in our animals, because these autophagic structures were filled with neuromelanin, we were able to remove this neuromelanin outside of the cell, and by doing that, we had an effect by reducing. We reduce the number of inclusion formation. We reduce the dopaminergic dysfunction. We restore the motor deficits, and we actually attenuated cell death. So by removing this neuromelanin outside of the cell.
So picking up on that, and perhaps Ariadna, I can ask you about this. So this is a sort of if you like, a well sort of researched area of Parkinson's disease. So, you know, simplistically, you can think of these diseases, including Alzheimer's disease, other diseases, that part of the problem is you produce too much of the wrong protein on the one hand. On the other hand, you can't get rid of it. So you can tackle it in two ways. You can either stop the production or you can increase the clearance. And of course, autophagy is like the sort of dustbin lorry, the garbage lorry, you know, it comes to get rid of things. So this strategy has obviously been talked out quite a lot about for getting rid of alpha-synuclein, not neuromelanin, just getting rid of toxic forms of alpha-synuclein, the protein associated with Parkinson's disease, in much the same way as tau for other diseases and such like. So how specific do you think you'd have to be, Ariadna, with this type of therapy? Do you think anything that upregulates this process, autophagy, of which neuromelanin goes out, but also probably alpha-synuclein, would be a good thing? So we don't need to be very specific about finding an agent which specifically gets rid of neuromelanin. We just need something that gets rid of, that upregulates autophagy and gets rid of anything in the cell, including neuromelanin. I mean, what's your take on that?
Yeah, that's indeed a critical question. As you said, like modulating autophagy has always been on the table as a therapeutic option for Parkinson's and many other diseases, and there are several regulators of autophagy that have been like proposed, and even some are on clinical trials. So it could be a possibility, although I think we have to be careful with this, because as you said, this might be quite unspecific. So we can start getting rid of everything, and we know that getting rid of everything is not good either. So I would be a little bit cautious, and I think we still have a bit of work to do on this field.
Yeah, because I've also been told, I've also had discussions on this, because of course autophagy is a pathway, so it starts at A, goes to B, C, D, and E, and obviously if you speed up autophagy at the beginning of that pathway, so A to B to C, but don't do anything for D to E, you could actually make the whole situation worse, because you just pile everything through a system that's already in trouble. So getting it to the right part of the pathway, and against the right agents, because as you say, if someone removed the garbage every 5 minutes, your house would be in a bit of a chaos, whereas you need to just get rid of it at a sort of at a regulated time. So is there any way, coming back to your point, Miquel, about, you wanted slightly less neuromelanin, so coming back to what I was saying earlier, one way is obviously to increase the clearance through autophagy, which you've done those very elegant experiments to show that. How about reducing its production? Are there ways in which you could block the production of neuromelanin without stopping it all together? Is there any strategies which you can think of which might be useful as a way of trying to look at in the lab and then use in the clinic thereafter?
Yeah, but basically we are working on different strategies. One strategy is because neuromelanin is formed from the dopamine that is not encapsulated inside vesicular synaptic vesicles. When dopamine is formed, it goes inside synaptic vesicles to be released. But the dopamine that is not encapsulated inside these synaptic vesicles gets into the cytoplasm and is oxidized into neuromelanin. So one approach that we are doing is that we are trying to promote the encapsulation of dopamine by overexpressing a specific protein that helps encapsulate dopamine inside synaptic vesicles. We are doing that by gene therapy, by injecting a virus with this gene. So what we see is that we help to encapsulate dopamine, so you have more dopamine inside synaptic vesicles, and therefore you have less dopamine in the cytosol, you have less oxidation of this dopamine in the cytosol, and you have less neuromelanin. And we see that with this approach, we are able to reduce the levels of neuromelanin formation and accumulation, and we have an attenuation of cell death and pathology in these animals.
And do you think that could go to, I mean, obviously that's a very particular manipulation in the lab using gene therapy.
Yeah.
Do you see this, do you see a path for this to the clinic? So could this type of approach be used in the clinic? Are there agents which we have, or gene therapies which could be developed around this? Because I can see the strategy really is to try and keep dopamine where it should be, and try and stop it getting out to where it shouldn't get.
Well, that could be, but this implies gene therapy directly into the brain, and this is a factor actually that PD patients, they have a decrease encapsulation of dopamine, so that could help. But we are also trying other non-invasive approaches, like for instance, what is called the transcranial focus ultrasound, by applying ultrasound. Some of these ultrasound techniques are already being applied to patients, for instance, to produce some kind of lesions, to reduce tremors, for instance. But in our case, we are using low frequency ultrasound that you actually do not kill the cells, but we are trying to degrade the neuromelanin and to help the cell eliminate the neuromelanin. And also this ultrasound approach is combined.
So this is a bit like trying to get rid of kidney stones.
Well, more or less, yeah, more or less.
So this would be like a nerve stone.
Yeah. So that could be easily translatable to humans if it works, because this technique is already in place for the human brain. So that we are trying to do that.
That's a very clever technique. I thought what you were saying was you use it because people have used this to slightly open up the barrier between the blood and the brain, so things can get in temporarily. So I thought you were going to use it for your gene therapy, but for actually breaking down neuromelanin, that's a very novel approach to try to.
But we are also trying to combine also with this transient permeabilization of the blood-brain barrier, because of course, if you help the cell release neuromelanin outside,
Yeah.
Then you will have neuromelanin at the extracellular space. So we want also to help the inflammatory system to get rid of this neuromelanin. So we may combine this ultrasound approach also with this permeabilization, transient permeabilization of the blood-brain barrier.
Lovely. Well, I think we've sort of coming towards the end of our time. I just want to thank you very much for this very interesting discussion on something which I would say has been slightly overlooked in the field of Parkinson's disease. I mean, as you say, it's been around for 102 years, the observation, and people have talked about it, and it periodically pops up in the literature, but it's never really been systematically addressed and approached. And I think you're to be congratulated on taking this on in a very courageous way and developing models which now open up all sorts of possibilities for understanding how neuromelanin can cause or lead or promote Parkinson's disease. And the beautiful thing about research, as you've both so eloquently demonstrated, is the excitement of discovering things. And as you discover things, many more questions appear which you'd never thought about in the first place. So there's always this, I always remember when I started research, you sort of think, well, I've only got 3 years of ideas. At the end of that, I'd have run out of ideas. But actually, as you start, things start to appear as you go along. And you now have this wonderful system where you can start to interrogate all these questions. So I would like to thank you both very much for a very clear hour's discussion on a very, very interesting topic. Congratulate you on the model you've developed, the interest you've generated. I would like to thank our audience for staying with us for this hour, and for people who subsequently listen to this. I would like to thank WPC for setting this up in the first place. And I would just like to tell people that there will be another one of these. So as Eli said at the beginning, there will be every other month. So I think there'll be 6 this year. And the next one is on Tuesday, the 23rd of March. It's at 4 p.m. Eastern time. So whichever time zone you're in, you have to work relative to that. And it's David Finkelstein and his team, and they are talking on something which some of you may have heard about, which is COVID and Parkinson's disease. So they will be talking on aspects of coding and Parkinson's disease. So thank you very much. I hope you found it interesting. Do go and read the blog, listen to this again, and keep tuned into the WPC and all the exciting work that will come out. And don't forget, next year, we're all going to go and see Ariadna and Miquel, because the WPC is in Barcelona. So we'll see you all there. Okay, thank you very much. Bye-bye now.
Bye-bye.